There we go. Better? Perfect. Oh, they missed all my intro on the recording. That was only for you, so it's special. Okay, so yeah, so we're talking about other samples where there's a lot of host DNA. Then 16S is really only going to target what you're after. And so what, in comparison, is obviously what we're going to talk about a lot today is shotgun metagenomics, right, or metagenomic sequencing. And that's essentially sequencing now all the DNA in the sample. And so that obviously has lots of advantages. One, you don't have to pick a primer, right? So hopefully we're getting really good amplification across the different DNA. So you don't have to worry about variable regions. You don't have to worry about different types of primers and that biases. You can identify all microbes in one. So we're not just getting bacteria and archaea or having to sequence, say, ITS or fungal. You're going to get, if there's fungus in there, you're going to get fungal sequences. If there's viruses in there, you're going to get virus sequences. The downside to that, of course, if there's host DNA in there, you're going to get host DNA. So that means if you're doing something like the saliva microbiome or skin microbiome, and you're doing metagenomics, you're going to have to filter out a lot of that host DNA. And that means, obviously, you're going to have to sequence more to account for losing all of that data. So typically, say with saliva microbiome, we're talking 90–95% of that would be human DNA if we're doing metagenomics. Maybe sometimes you get lucky about 80%, but still a large portion of that's going to be human DNA. You got to remember, human cells are bigger. You're going to extract those, and they're bigger genomes, right? Yeah. Question over here. Yeah. Yeah, it's something that I haven't personally ventured into, and the IMR hasn't ventured into it all. There has been definitely some approaches to trying to select for more microbial DNA over human DNA. They work somewhat. So we sort of looked at it a little bit. The issue a bit becomes is that these kits tend to be a little pricey. And so often if you start to look at the price of the kit for doing the selection, and then you compare that to just how much it would cost to do extra sequencing, it might not be actually that far off. And then the downside to doing the selection is you always have to worry about whether you're biasing your microbiome sample, right? Whether you're selecting for microbes, but maybe particular microbes. So there is a bit of an issue. There's been definitely a bit of success on the other side where we've seen it, where people will try to remove microbial DNA to do human sequencing. And so they work fairly well there. But the biggest problem is we do worry about biases with the microbiome. I wanted to mention too that definitely feel free to ask questions during the lecture if you like. I don't have a ton of slides, so we can take questions throughout. Okay, and the other big advantage that gets touted a lot with metagenomics is better taxonomic resolution, right? So with 16S sequencing, you'll get genus level, maybe the occasional species level. And in theory with metagenomics, especially if you have enough sequences, you get down to possibly species more often and sometimes down to a strain level or the ability to track a strain if you have multiple samples. So that's another nice advantage. And then the other big one is what we'll talk about this afternoon is that it provides not just about information about who is there, it's also like what they're doing, right? So you get sequences coming from those genes. We can functionally annotate those genes, and we can get an idea about not just about the taxa, but also the genes and the pathways that those microbes encode. And so that's a huge advantage as well. And lower hugs, we'll be talking about, we can also possibly reconstruct genomes. So actually forming the original genomes through assembly and binning. And the only major downside is cost beyond the host contamination issue, right? So we have to sequence a lot more to do this, usually in the range of 5, 10, 20 million reads per sample. Some people will even do more. They'll do 100 million reads per sample, but obviously that's a lot more than 16S sequencing. And there's something else around that. Oh, some people have sort of dabbled, I guess, with shallow shotgun sequencing. It's something you might have heard about. Shallow shotgun sequencing typically, I guess, would be maybe in the range of 1 million reads or less, seems to be the definition around that. And so that sort of became popular Some people think it's fine, whether it's better than, some people argue it's better than 16S. And I don't really have a strong opinion about it. I feel like probably if you're going to do metagenomics, it doesn't probably cost that much more to get a full metagenome, right? So we're talking about shallow metagenomic sequencing costing you maybe $25 less a sample. It's hard to say. Metagenomic sequencing does vary a lot. We're talking sort of in the $100, $150 range per sample compared to 16S. Although we have seen a drop a bit across the board. Yeah. Did you say 3 or 10? So the question is like, how much sequencing do you need? Yeah. I think people's comfort zone is probably in that 5 million reads per sample and up, typically. I think a lot of labs will do around 10 million. Our basic one at the IMR is 7 million. That's on average, of course, right? So you're going to get some samples with less, some with more. You know, I always like to say though, back in 454 days, people were doing metagenomics with like 500,000 reads, right? So environmental sampling, which I don't think there's too many here. Obviously, you're doing a lot more depth. Definitely see people doing 100 million reads per sample. Seems most people are pretty comfortable with 10 million or more in that range. Absolutely. Yeah, absolutely right. So that's if we're talking about stool where you're going to get basically 100% microbial reads. If you're talking about some other site where you're tossing 80% more of the reads, then you're essentially trying to account for that. And people will do less sometimes. Yeah. So we did have a paper a few years back where we did look at biopsy samples in Crohn's patients. And so with that one, we wanted to look at the metagenome of Crohn's patients in a colonoscopy collected sample. And so with that one, we did it was around 100 to 150 million reads per sample, and then end up tossing, 98% of the data. Well, we didn't toss it actually, so it was a really nice paper. I usually talk about, I'm not going to talk about today. We took those reads, those other 98 million, and then we actually did genotyping on the host. So it wasn't a waste. You just used it for your host genotyping. So you can sort of factor that in too. And one thing we've been sort of barking up the tree about lately is that we've been looking more at people have done human genome sequencing or human genotyping, or human genome sequencing. And so they've done sequencing and they've tossed, any reads that didn't map to the human genome. But in theory, if there's microbes there, they would have microbial reads that they just filtered out. And so we're starting to get a couple of projects going at that angle. Yep. Okay, so pretty straightforward here on the taxonomic profiling, which we're going to focus on in this lecture a bit more. We'll save the functional stuff for the afternoon. And you might be thinking, if you don't have metagenomic data and you're only got 16S, you're like, can I just tune out the whole day? No, you don't want to do that. It's nice to know about other technologies. And then two is, sometimes I would have a separate PyCrust lecture. I will mention PyCrust in this afternoon's lecture, and that will give you some idea of like, if you have 16S sequencing, you might be able to get some functional information. So it'll come back around. Okay, so this really basic thing of, we have raw data coming from metagenomic sequencing. Obviously, the reads are coming from all the organisms mixed together. We don't know where they're coming from. And we're trying to essentially get the similar data that we would have for 16S data. We want taxonomic profiles, and we want their relative abundance in that sample. Seems straightforward, right? This is really what we're trying to do. And the take-home thing is that it's still pretty hard to do this. So the challenges are quite a few from different perspectives. So one, as I just said, the reads are randomly assorted. We don't know where they came from. The reads are usually fairly short by themselves. So we're talking usually 100, 150 base pairs as it's coming off an Illumina sequencer. Now that could be changing in the future more as we see more full-length sequencing and adding that into metagenomic samples where you can get really long reads. And so we will see more and more of that upcoming, but still you're not going to get the same depth with long reads, and so there's a trade-off there as well. So usually we're still talking fairly short reads. And we're sequencing at a depth, say if we're talking about this 10 million reads, that in, say, a human microbiome, definitely there's hundreds of different organisms. We're not going to get full coverage of the vast majority of these organisms, right? So even for the most dominant one, we still might have spotty coverage on its genome. For the things that are more rare, we're going to maybe get a few reads, 10 reads, 20 reads to some of these organisms, right? So we're not going to have its full genome. And so that makes assigning taxonomy to just a few reads much harder than, say, a whole genome. Lateral gene transfer will come into play here. So if we're getting random samples of reads, we're not talking about nice universal marker genes all the times. They're hitting any spot on the genome. That could be repetitive regions. It could be genes that were recently horizontally transferred. And that means, hey, this looks like this thing, but it was horizontally transferred. We don't know that. Then it's going to give us a false signal, right? So that's problematic for sure. And one thing we can't forget about is computational time. So we're talking about very large number of reads, 10 million reads per sample. Typical experiment, we're talking 50, 100 or more samples. That's quite a bit of data in your hands. And then we're comparing those to very huge databases. And we'll talk about that actually in just a few minutes. So computational time comes into play here with, hey, can we just, blast sequences? It's like, no, you'll wait 10 years for it to be done. So we have to take shortcuts to do that and get results in a reliable amount of time. And this is a bit of a throw out to, not forgetting other biases. So we're going to focus on bioinformatics, but I wanted to bring up this, which kind of comes back to Marie Claire's talk a bit yesterday, and is a paper that we had come out last year. It's just a review looking at all the different types of biases that exist in a microbiome experiment and sort of cataloging that. And so this figure is just showing everything from sample collection through to DNA extraction, library preparation, and the different major steps of bioinformatics experiment. The things in the orange boxes are different things that can account to a bias resulting in different taxonomic profiles. Super depressing, super scary, but the good thing is that you can control some of these, right? So the first part and even the bioinformatics, if you do the same thing over and over, at least it's not causing a bias across your experiment. And then some of these obviously have bigger biases than others, and that's what the review goes into. So DNA extraction, Marie Claire really covered that well. Big component to creating bias. Things like GC content there and library kit, construction kit used to be a bigger problem, not so much a problem now with sequencing. And so the review sort of walks through some of those and suggests things to watch out for. All right, so if we come back to bioinformatics steps. So let's get into some of the nitty gritty. So if we talk about the initial bioinformatics steps, the nice thing about coming second is a lot of this was covered nicely by Diana and Rod yesterday, is that many of those initial steps are similar to 16S studies, right? So those include things like demultiplexing, lane merging as it comes off the sequencer. Quality filtering is obviously a huge one here. And so me just putting quality filtering is a bit of a thing that I don't have to dive into. It was covered nicely, but things obviously like trimming reads, discarding reads, that really comes into play with metagenomic data, because you don't want to feed, low quality, problematic reads into some sort of a taxonomic assignment tool. And that tool, it's hard to say what it's garbage in, garbage out will help it. So you give it something, it might say, there's zebras in your metagenome because, it's some repetitive region that is also in zebras, right? Definitely you can, I remember we did ocean studies in the past and we found cows in the ocean. There's no cows in the ocean as far as I know. It's garbage sequences. Maybe the taxonomic profiler could have got rid of them. Hopefully they would do some filtering to, account for that. But the reality is they're sort of assuming that they're getting microbial reads that are fairly high quality. We talked about stitching reads a little bit. And so usually with paired end sequencing, which is typically done with Illumina, unlike 16S or amplicon sequences where sometimes they'll overlap or the majority will overlap if your amplicon's small enough, and you would stitch those together. With metagenomic sequencing, that fragment of DNA that gets sequenced is variable, right? Because you're randomly chopping things up. There's some selection a bit on size, but they'll vary. And so that means when you do a forward read and a reverse read, they'll often not overlap, right? Because you'll have, say 1,000 or a 500 base pair fragment. You do either end and they're not going to overlap. So typically we don't stitch reads because they just won't stitch together successfully. So typically not done. What do you do then with paired ends? Well, it depends, of course, but often what will happen is at some point you'll end up just taking the forward reads, taking the reverse reads, and you just smash them together. Like you just concatenate them together. And essentially what happens then is you're counting things twice. In the occasional time, some mappers will take the forward and reverse reads and use that information to properly map. But if not, then with many tools, you end up just concatenating the reads together. And then removal of unwanted host associated reads is probably the first major step that's unique to metagenomics, right? So this is where we're trying to get rid of host contamination. So everything to that point is really similar to what you'd see in 16S studies. So with this, contaminant reads is obviously what we just talked about before, right? So we're looking at, for a lot of these experiments, host would be the contaminant reads I'll refer to here. So you would typically run something like Bowtie or BWA, which are mappers, and you would try to map all of your reads against the human genome. And typically in our lab too, we will also map to the phi-X viral genome. So phi-X is a typical spike-in that's used in every Illumina sequencing run. That's how they determine the quality of your sequencing from an Illumina run. Illumina, the machine will filter that out, but we've found that sometimes reads will, get missed and they'll still be included. So usually those will be filtered out. So we found it's good practice to include phi-X in our mapping step just to make sure that those reads don't come through and cause some problems. Yes, John, in the back. Yeah, that's a good question. The question is around, those human reads. What do we do with the ethics of that? Because in theory, those are, human genome reads. And what I've seen in the past is that people tend to gloss over this in REBs currently. So they say we're going to focus on the microbiome. We're going to do metagenomic sequencing, and then we're going to take the human reads, and we're going to take the microbial reads, and so it has nothing to do with human genome stuff. And they sort of gloss over this fact that they would get human reads at the same time. That's in practice what I've seen done. I don't know. I'm not an ethicist, so I guess in theory, if you're just discarding it and never going to look at it, maybe ethics doesn't come into play. It seems like something that should be included in ethics. And obviously, it's probably a bigger deal in things where then you do have significant host DNA, right? So a skin microbiome, oral saliva, vaginal even microbiome, you are going to have significant number of human reads. So maybe other people do a better job than what I've seen, and they'll include in their AB. I don't know. Yeah. Right. Yes. That sounds good. I mean, that sounds like a good plan to sort of treat those as something that should be a bit more secure. And I think what I have seen is, I think this comes into play probably the most number one thing is usually with most microbiome experiments, you're going to upload your data to the SRA or the ENA, right? To a public repository in many circumstances. And so if you're doing that, probably a good idea that the reads that you upload aren't just the raw reads. Maybe you're going to take this step here where you've removed the human reads and upload the microbial reads, unless you have, proper REB to upload the human reads as well. Yep, question in front. Yes. Yeah.
So maybe it's also necessary to, for example, only to discuss some
Just to be clear, you need to do 6 sequencing on top of the metagenomics. No, no, definitely not. So typically, most people are doing metagenomic sequencing will just do the metagenomics. Now, a lot of people will do 16S sequencing first because it's cheaper, and then they'll maybe do all their samples, and then they might follow that up with some metagenomic sequencing after. But it's thought pretty much on the whole that metagenomics does a better job than 16S because of the things I mentioned before, the primer biases, better resolution, things like that. The fun fact is that if you try to get those to agree, 16S and the metagenomic profiles, yeah, they won't. They won't agree very well. They won't actually, because also taxonomy is a problem there, right? So we're talking about Silva taxonomy is actually just fundamentally different than say NCBI taxonomy or the GTDB taxonomy, like at that level. But then you also get differences just because of potential biases and the tools we use. It's uncomfortable, but that's the reality. So to the answer, the quick answer is no. Typically, if you're going to do metagenomics, you would just do it by itself. Okay, yeah, so that's on the mapping side. And if you're thinking about, parameters for bow tie or something, you want to be fairly lenient with that mapping, right? You might as well trim off a few extra reads that you're not sure about that might map to the human genome and discard them as opposed to keeping them later, right? Because you don't want some reads are like, yeah, maybe it mapped to the human genome, but we'll put it through Kraken or Metaflan and see what it spits out. We want to try to minimize as much as possible. Okay, so let's get into sort of how we then talk about, what we do with this data. So this is from another a review paper in 2021, covers lots of different things. But essentially, this covers sort of maybe 3 major categories of what you can do with metagenomics data. We're going to focus on the A panel here, where we're essentially, can't see my mouse. Well, it doesn't matter. You probably can't see it back there anyway. The panel A, what we're typically doing is we're mapping our reads to some reference databases, either a taxonomy database for getting taxonomic profiles or functional databases for functional profiles. And we're coming up with this nice table, kind of like an OTU or ASV table with 16S data. That's what we're going to focus on entirely today. In C, metagenomic-based genome assembly, this is what Laura Hugg's going to talk about with this idea of how do we take these reads, assemble them possibly, form contigs, bin them, and put them back into essentially the cells or organisms that they came from. And so Laura will talk about that approach, right? And they're really complementary. The nice thing about this approach is it's raw reads. In theory, maybe get pretty good taxonomic classification of all the organisms. The downside of a metagenomic genome assembly base is that it will assemble obviously the more dominant organisms better. And so obviously you'll identify those really well, but then what about the chaff at the end? You may not do as good a job. And then I put this up, you don't see this as much maybe, but definitely was popular in the past and still, is really good, is this idea of mapping reads to genomes to look at, coverage of certain genomes in a reference database. This used to be called fragment recruitment. It was pretty popular. And sometimes what you're looking for is how well a coverage you're getting at. We actually do this a little bit with our blood microbiome, where we're really trying to show, you can imagine if an organism's there and you really want to show it's there, we should have pretty good coverage, not just one spot in the genome. And it also can give you an idea about if you have a reference genome and you're comparing it to something in your sample and you're missing a region, like say this region right here, then maybe that means that your particular strain in your sample, doesn't have this region, right? And so you can start to do almost like a comparative genomics approach using that information. But like I said, we're going to focus primarily on this raw read-based approach, mapping to reference databases.
Sorry.
Yep.
Clarify, kind of read.
Yep.
You mapping the reads to the genome of interest, like reference genome.
Yep. A reference genome, especially people do this if they're particularly interested in that one type of species, right? So just take whatever species you want, say Blastocystis or something, right? Which is kind of an interesting genome. You might want to say, can we find new Blastocystis genomes? And you can use mapping to say, okay, what regions of this genome is covered? Do we possibly have a new strain or something? Or is this a variable region within this genome that gets excised, and so it may represent like a genomic island or something? So yeah, it's much more targeted usually on a per species level that you're interested in. Okay, so now we're going to drill deeper into our reference-based approaches, and then I'm going to split these into sort of two main approaches within reference-based. So one is what I call the all reads approach. I still don't have a better term for this, but this is the idea of you're trying to map all of your reads to some sort of reference databases, and you're trying to essentially classify all of those reads as much as possible for taxonomy. So this sounds good. This is kind of what you would think about when you're doing this. It means similarity search is computationally intensive, right? So you're trying to map all your reads to a very large database, and we'll talk about that in a bit more. The downside, this is what I mentioned before, is that because we're trying to annotate all those reads, some of those may not be good representative markers. They could be repetitive regions, they could be LGT events. Many reads won't map at all because you just don't have those things in your database as well. There might not be in homolog. But the bonus is that the nice thing about this is you get taxonomic classification, and you can also then dig back to say, okay, these are the reads that belong to that taxonomy because we assign taxonomy to them. The other major approach is sort of a marker-based approach. And so this is analogous to almost like a 16S approach, is where we use particular markers. So we just filter our data and we only pull out certain genes that map to certain markers, and we use those to determine the taxonomic composition. The downside is it uses only a subset, which is an advantage computationally. It means these things are fast because you're essentially just filtering for certain markers and assigning taxonomy. But of course, this is very dependent then on the choice of markers. Which markers do we choose? So kind of back to 16S land. In the past, we have seen people pull out, you could pull out 16S sequences, right? So this is why you would typically not do a 16S experiment. You could pull out your fragments mapping to the 16S gene from your metagenome. They're going to be in there. And you could use those reads and put them right through QIIME and do that. You could use other universal markers and use existing bioinformatic pipelines. We don't typically see that done much anymore because I think it's sort of seen as, you've done a lot of sequencing, you might as well use it all, as opposed to just pulling out those markers. But it is something you could do. And then the more popular methods are using multiple markers. And so things like MO2Use uses 10 universal single copy genes. So not just 16S, but other universal markers. And then definitely the most popular, I would say, and this is clade-specific markers, which I'll talk about in a second, and that being Metaflan3. So from here on out, I'm just going to talk about Metaflan3 a little bit, and then we'll come back to an all reads approach. So Metaflan3 uses clade-specific gene markers, which I'll explain in the next slide. And these markers are derived from a large genome reference database that they've previously created. This comes out of Curtis Huttenhauer's lab. And in their most recent version, I think it's around 1.1 million different markers are used to taxonomy classify. They get a pretty good resolution, sometimes down to the strain level. And the really big advantage is that you can do this essentially on a decent laptop in a few minutes per sample, right? So you can throw a sample at it, it's going to do taxonomic classification. No large computational requirements are needed for this. This is just a figure straight from one of Curtis's slides, and so this is just getting at what these markers are. And so the panels on the top is just showing essentially, say, a gene. It doesn't even need to be a gene though, but it has to be a region that are both within this particular clade. And so by clade, I mean this could be at the genus level or family level, and they're found within all of those. and it has to also be, unique or fairly unique to that clade, right? So as opposed to a universal gene that's found in everything, they're looking for particular genes that are found in a particular clade and only in that clade, as far as they know, right? The downside is as far as we know, because they're using, this is our universe. Of course, if you sequence more genomes, some of those markers might change. That's the downside. So that essentially what they do is they look for these markers ahead of time. They pre-compute this. They use their, an offline pipeline they call it Chocolflan. All of these markers are pre-calculated and figured out so that when you do your mapping, you're essentially just mapping your reads to these markers. And so that's a much smaller database than, say, something like we'll see with Kraken. And then it does taxonomic classification. Moving to an all reads approach, there's several out there as well. We're going to talk about Kraken and Bracken, but there's many as well, and you'll see that in a couple slides. Centrifuge is also quite popular, Keiju, and many, many others out there. There's many, many approaches. Most of these use what's called a K-mer-based searching strategy. And so this is where we get to, that computational bottleneck. We're not going to throw blast at this. It's going to be too slow. So these will use a K-mer-based approach, which I'll explain in a second slide. And they also usually use some sort of lowest common ancestor approach to assign taxonomy. And I'll talk about that in a bit more detail. That's pretty common across almost all these methods. And they just use different databases and different heuristics on top of the K-mer approach to speed it up even further. And there's lots of trade-offs between obviously speed and accuracy. So you might have heard about K-mers before. I think it may have even came up yesterday. I stole these slides directly from Robyn. Thank you, Robyn. And so K-mer-based approach is essentially all you're doing is you're taking a particular genome of interest, you're looking at its genome, and you're literally slicing up that genome in K-mer sizes. So in this particular case, this is 5 mers, okay? So you say your K size is 5. I'm going to use a K-mer size of 5. It literally will scan along overlapping regions, pull out all of those sequences. And you're thinking, why would you do that? This sounds crazy. And I'm not going to get into obviously the computer science of it. But once you do that, you can really efficiently then store those in a database. And you can then just look for identical matches to those K-mers down the line to say, oh, I have this K-mer in this sequence. It matches perfectly to this K-mer in this genome. And that is super quick. And then you can use that to scale up and you say, well, how many K-mers match between my total read and this total genome? And you can use nice databases to efficiently do that. You can collapse this complexity and speed up this approach quite well. The other thing that you can look at is what you'll often hear about is LCA, or lowest common ancestor approach. And so what this means is that because you're going to get K-mers or matches across multiple species, right? Especially for K-mers, obviously you're going to find lots of genomes with the same K-mer. So which one do you assign it to? So what you do is you start to say, okay, well, how many different things is this shared across, right? So at the species or strain level, it's found in, okay, E. coli for this particular, say, gene or K-mer. It's not found in actually this species of Escherichia, but it's found in Salmonella, and it's not found in Pseudomonas. And then you just basically say at what level then is the lowest part in this tree where this K-mer maps. And then you say, okay, it's at this branch. This is a taxonomy tree. So then you're going to say this is at the Enterobacteriaceae family. So that's lowest common ancestor, okay? So if we look at Kraken 2, it does that as its first step. So yeah, I'm going to walk you through this a little bit slower. So this is our query sequence. These are all the different overlapping K-mers. And then the maps from one of these K-mers to our taxonomy tree is shown by the different colors. If there's no color, that K-mer didn't map to any reference genome in our database, okay? So the first step is to do this LCA mapping, right? So you put those in the tree, and that's where we get our assignment for those K-mers. Great. Kraken 2 then does a next step, and it says, okay, let's examine this hit tree across the entire query sequence, because we have all these different K-mers. Like, what is it? What is it in these? So what it does is it then looks at basically all these nodes, and it counts up how many of those k-mers map to that particular node. And that's what's shown in this area, okay? And then it'll take its highest weighted RTL path and try to call this as that sequence, as this taxonomy. One thing that we've dug into quite a bit with Kraken, and I'll talk about this towards the end of the lecture, is that Kraken 2 has what's called a confidence cutoff, a confidence score. And what that means is it just says, if we count up all these different k-mers, there's 27, we have 4 of them at this part in the node that map to this. So that's 4 out of 27. So it gives us confidence score of 0.15 for this taxonomy assignment, right? Now, if we moved up the tree, we could add this 10 plus this 4. And so if we called it at this level, the resolution's not as great, right? It's higher taxonomic resolution. So we're going to lose some information. But the confidence score goes up because we have more k-mers mapping to this particular taxonomy node. Okay? And then the same thing, if we moved all the way up here, we would sum them all up, includes all of these. So we'd have 0.59. Now, Kraken 2 actually has a little nice option in there, just deepen, there are options of different things you can turn on. And so by default, the confidence score is 0, right? So we get basically this assignment down here. Okay, and we'll show you that that's probably not great. It leads to false positives. But if you up that to a cutoff of, say, 0.2, then that's going to be anything below that's not going to be included. And so we would call it at this blue node, a bit higher resolution, but with more confidence. The other thing is that you'll often hear with Kraken, it's super confusing, is that you should run Bracken after Kraken. nice terminology. And I'm not going to go deep into this, but it's this idea that you can imagine if you sequence more and more genomes, right, and you try to map these k-mers as you add more genomes, it's going to start to push a lot of those k-mers up the tree because it's going to be shared more. And you almost lose taxonomic resolution. And so Bracken is this approach to try to account for that, to say, well, look, we have these reads assigned to this one read. We've pushed this up to a certain level. Bracken will try to actually push the resolution back down to improve the resolution based on multiple reads hitting a particular organism. I'm not going to explain it all, mostly because 1, I don't actually understand all of it. But 2, it's just too complicated to get into here as well. So, but yes, so usually if you're going to run Kraken, you're going to have to run Bracken after it to improve your resolution. That's the take home there. Okay, so those are the 2 major approaches: general approaches, all reads approach, marker-based approach. And then the big question is, okay, so I have my data, like, I don't care about all the methods. I want to know what method to use. So, which one should you use? This can change over time, especially as databases grow. New tools are released. It depends on how we assess this, whether we're using mock or simulated data sets that can cause, one to look more accurate than others. It depends who you ask, as I'll walk you through a couple of papers. And it also depends on some underlying approaches about really what you're trying to trade off for, precision versus recall, as we'll see. So lots of people have done nice comparisons of these. This was a paper, I don't know how it got in Cell, to be honest, but anyway, it was a Cell paper in 2019, good for them. They looked at 8 simulated samples across a whole bunch of different tools. So on the bottom, you'll see, as I mentioned before, there's a whole bunch of different tools. Those are those tools. The ones in red are marker-based, and the ones toward the left are sort of all k-mer, all reads approaches, we'll call them. I'm not sure why the orange ones are orange. I think it's a translated search in a protein database. I think that's why. So this looks okay. AUPR is essentially the area under the, I won't guess, but essentially it's a precision recall curve. You can refer to it almost as accuracy, so higher is better. They look all pretty good, except for centrifuge for some reason looks crap. Also, the marker-based approaches don't look very good in this comparison paper in 2019. So you'd be like, okay, bad marker approaches, let's use something like Bracken. Then another paper came out from MOTUs. These are the authors of MOTUs too. And they showed that, their method did really well. So this is precision recall. That's theirs. Red is MOTUs. Top right is really good. This is absolute error. So lower bars here are good. So they look way better than their first version or Metaflan 2 or Kraken. And this figure here just shows the absolute number of species and how much they're off. So if you're at 0, that means you're over or underrepresenting too many. So you're like, wow, Nature Communications paper, MO2 looks awesome. They have like great precision recall. And then Metaflan 3 comes out, 2021. They compare their tools against other tools as well across different body sites. This is really hard to see, so unless you're looking at your own computer. But essentially these little tiny red bars up here are almost at 1. That's Metaflan 3, and they're doing better than the other approaches. So you may have seen this thing now where it really depends on who's doing the analysis. It seems to maybe be, if you're launching a program, your tool always does best. Imagine that. So, even though there's lots out there, we decided, hey, let's, dig in this a little bit deeper. So this is data from our preprint that Robyn led, and she's going to talk about this at the conference in Canmore in a little bit more detail, but I thought I'd cover some of it. But essentially in this one, we scaled down quite a bit. We're only comparing Metaflan 3 versus Kraken 2. The reality is those two are probably the most popular methods out there. Popular doesn't always mean best, but the reality is these are the tools that are cited a lot and are probably the two big divides, a marker-based approach versus this all-reads approach. And we wanted to really look at two major things, the effective database size and some parameter selections within these tools. And so what I'm going to show you first is these are just different databases. So at the bottom, they're the smallest. So Mini Kraken 2, this is a default database sent with the Kraken tool. It's thought to be small. It runs on your, say, your computer, your local computer, doesn't require much memory. And then these scale up to much larger databases that have more and more genomes. So this is the database size in gigabytes. This is the number of taxa in those databases. And this is showing the coverage of those databases to the simulated data sets that we used in this particular analysis. Okay, so it's just sort of setting up these different databases. One thing to keep in mind is that when we talk about Kraken 2 databases, the size of those, so say we talk about a 500 gig database, literally what Kraken does is loads that whole entire database into memory. And that's how it's so fast. So that means if you have literally a 500 gigabyte database, you need 500 gigabytes of memory on the computer you're running on. Your laptops have, what, 8 gigabytes, 16 gigabytes? So you're not going to be able to run anything except for Mini Kraken 2. You're talking about 1.5 terabytes of memory. There's a few servers out there. We have one. You can come talk to me. But there's servers out there, but obviously you need really high performance computing, right, to do this. And so that's where this computational requirement really comes into play. But anyway, let's talk a little bit about the major differences. We're at 10 o'clock and I just have 8 slides. Is that okay if we just go a little bit over? We started late. Will, is that all right? Sweet. So this is, I know it's hard to read, but essentially it's increasing database size, and then it's human associated reads. These are real samples and environmental on the right. And this very first bar is 16S data. And what we have on the y-axis is number of species. All this is just showing is the really big take-home message here, and this is why we had known this for quite a while, is that Metaflan 3 tends to underrepresent the number of species. So if you just count the number of species in a sample, not that 16S data is right here, but just giving you an idea of where we might think the number of species is. In human associated, this is on average 64 species per sample. And if we move to Kraken with different databases, there's 2 bars for each one. The lower bars, this is with that confidence cutoff of 1. So we moved it all the way to the most extreme of robustness. But without that cutoff, so just using the defaults, is what you would get in these other colors. So you see a huge number of species, right? So you run Kraken 2 on your human associated samples, you're in the thousands, multiple thousands of different species. Metaflan 3 is like, you got, 100. Massive difference. If you do use a very stringent cutoff, you can drop that down. And with environmental, we see that even more extreme. So these are on log scales, just to take note. Environmental, metaflan-3 tend to really crap out. This is where we got really frustrated. We had soil samples and we were like, metaflan, no offense, I mean, it's a great tool. I like Curtis. But you'd get like 20 organisms in a soil sample, and we just knew that wasn't right. And then Kraken would spit out, again, thousands of different things. So we wanted to explore, between 0 and 1 for Kraken, what does that look like? And so for this, we used a whole bunch of simulated samples. Robyn, I'm not sure how many, and John will take your question in a second. How many samples? 400 simulated samples, yeah. Some of them are from 2 previously done ones, CAMI and CAMI2, and some other mock communities as well. Yes, John. Yes. Yes, absolutely. So Metaflan tends to do a really good job on human samples, definitely adequate. And then on environmental, it really, really craps out. And that's just because I thought that it has really good, there's just more human, obviously, microbes out there. There's a lot more unknown diversity in soil samples. And so you can imagine not getting the right markers in a very diverse sample. And so Metaflan just goes, there's nothing here for me. Yeah. And they also tend to focus, on, I would say, more human samples too. So they did a lot of their validation that way. I mean, you can still use it on environmental, but it just tends to be less. Yep.
Yep. This an easy fix? They just need to update the database a little bit, since Kraken can't do the job. So you can use this to inform how to update the database.
No, it's not. And the reason is that if we think about a database sort of saturating, so for human microbes, if you looked at our like rarefaction curve of how, if we sequence more and how many new species we get, we're sort of plateauing there, right, with human microbiome. And with environmental soil samples, like it's still going way up. And so we're not plateauing. And that just means that we can take the biggest database we have, and you would still go into the environment, and the markers are based on trying to find them all for this organism to say it's there. And it can't do it because this organism we don't have any real close relatives to it. Whereas Kraken says, okay, I have some similarity here, so I can do it maybe a bit better job. It's also because Metaflan, as we'll see, tends to value precision over recall. Yeah, Robyn, do you want to clarify on that? Yep. Yeah, that's a really good point. Thank you. So just for people that didn't hear, basically a lot more complexity in environmental samples. The chance of hitting multiple reads to a particular taxa in that is less because you have a lot more diversity and less sequencing depth. And so Kraken only needs one read to say this thing's here, whereas Metaflan requires multiple markers. Yep. Yeah, Robyn's here. She can take all your questions. She did a lot of parameter tuning on, so we talked about, I just showed Kraken parameter tuning. We tried a lot on Metaflan too. Didn't really actually affect too much. So this is just showing essentially that confidence threshold sliding from 0 to 1. So remember, 0 would be the default and 1 would be the most extreme. And so you see this sort of classic trade-off between precision and recall. So precision is true positives over true positives plus false positives, right? So our ability to robustly identify that thing. And recall is like how well do we do a good job at covering all the organisms in that sample. And so we see this classic sort of trade-off, and mean F1 score is essentially harmonic mean of recall and precision. So taking both those into account, fairly normal accuracy measurements here. And so we see that, at 0, where we get fairly poor precision, fairly decent recall because we have a lot of species, right? But overall, probably not the best choice based on all these similar data sets. We see, if you go to about half, our precision starts to basically plateau. Recall doesn't really take a hit majorly until the end. An F1 score is essentially maximized around 0.6. Just to give you an idea of proportion of reads classified. So again, if you increase that confidence threshold, we're going to get less reads annotated with Kraken. And so that's what's shown in that first panel, where you start to drop off significantly in the proportion of reads covered. This is just 2 different measurements. On the right is diversity and Shannon diversity. So looking at alpha diversity, you see not a huge difference except at both extremes of that tail. And then this is just a similarity in number of species identified. So this is essentially the 0 line here, right in the middle. There's no line, but 0 is here. If we're above that, that's saying we overestimated those number of species. Remember, this is based on mock simulated data sets, so we know what the truth is. So you can see this is on a log scale. So we're like on the order of hundreds or thousands more than we should by just counting them. And then once we get to really high confidence, we start to fall below that, where we're maybe underestimating that. So again, that trade-off is somewhere around this mark. I'm not going to show you the rest of it. Unfortunately, this is just using one database, and depending on what database you use, this, finding the maximal cutoff, of course, changes. But something in the middle looks kind of good. Okay, and then my last slide about this is comparing, I guess, Kraken 2 versus Metaflan 3 using different databases and using different cutoffs. And so what's shown here is the proportion of reads classified, just as I defined it before. And this is using different confidence thresholds, using the largest database. This database is our over 1 terabyte size database. Notably, this is the middle one. So this is a Kraken 2 database we built based on Metaflan. So this is really where, if we compare apples to apples, if we use the exact same number of genomes that Metaflan uses, and we say, okay, let's build a Kraken database, that's this middle one. So we can sort of say this is a fair comparison, I guess. And then this is Metaflan 3 based on that same database. So we see number of reads, obviously less with Kraken. This is estimated proportion of reads, just to let you know. And then we see, fairly decent coverage here. And if we look at mean F1 score, if we jump over to the right, we see that, with a confidence threshold of 0, definitely we're losing out with Kraken, where Metaflan does pretty good. But if we increase somewhere to that 0.5, 0.6 range, we maximize our F1 score. And then if we look about, again, the number of species, this is similar to the other one with our 0 line. We overestimate the number of species when we have a confidence threshold of 0. But once we're at that 0.5, 0.6 range, it seems to be closer to that. So, okay, I went a bit deep there, but it's new results. The preprint's out there. And so what does that mean? Metaflan's 3, great if you want fast, low computational requirements, definitely in a human-associated thing. You shouldn't feel bad about using Metaflan 3. It does a pretty good job. Simple bioinformatics setup. The database works by its default. The parameters work by default. Good precision. So the things that are there, you're probably pretty confident in. You're going to suffer maybe with some recall. You're going to miss some organisms. Kraken 2. Good for human and environmental data sets. So I think if you're doing anything environmental, I think you really got to go probably the Kraken 2. Unfortunately, out of the box, it's kind of a pain. The default databases aren't really big enough, so you're going to have to build your own. You can grab the ones that we used in the paper from us if you have it off of Dropbox. You're going to have to change probably that confidence cutoff to something a bit higher than 0. And the big take-home message there is use the biggest database you can find that you can fit on your machine. So for us, we sort of try to update as new genomes come out. Okay, and so with that, what is this? I will save this for later so we don't get behind, because this is just another reminders. I can talk about this after, and I will talk about, no, I'll just talk about it right now. Let's get over it. It won't take very long. So this is just a quick reminder. I like to remind people that when we're counting these things, we're talking about relative abundance and not absolute abundance, right? So it just means we have to take into account the different proportions of taxa. And that just means that if we have a sample A and B, this is just showing from a paper we published, that can mean, 3 different scenarios depending on what the absolute abundance was. So if the absolute abundance was the same between the samples, then it's great. You can make those comparisons. If the absolute abundance is larger than A, that means that this taxon is actually going to look like more. And if we have an absolute abundance of, at the right level, they could actually look to be the same. And so without absolute abundance, you're left with relative abundance, and you only talk about relative abundance changes. This mostly comes into play if you're talking about, say, pathogens or anything where you just try to say, hey, we have more of this thing. You actually don't know if there's more cells there. You just know that the proportion within your samples has changed. And then the last thing I just want to mention, because this will, I guess, play into the lab, is that we'll be running through a tutorial. I mentioned Microbiome Helper. It's essentially a set of bioinformatic standard operating protocols. So we have one for 16S. We have tutorials as well. We have some custom scripts there, and we actually have sequencing protocols as well. So it's a pretty good resources that you can check out. There's essentially a wiki there, and on the right-hand side, you'll see links to these protocols, and they're literally, I don't want you to just copy and paste commands, but the commands are there with all the options, so you can follow those, and that's what we use in our lab. So when there's an SOP, that's essentially what we're using currently within our lab.